So I'm going to talk about how the maternal genome, through epigenetic-based information, can contribute to developmental control, in particular growth control in mammals. So first, because we are dealing with information that can be transmitted from one generation to the other, of course, we are talking about germ cells, the oocyte and the sperm. And actually, what kind of DNA methylation-based information do they carry at the time of fertilization? If you represent very schematically the genome into a genome compartment, so here would be a transcription unit, a gene with exons and introns, intergenic sequences, transposon repeats, and you can see that the DNA methylation patterns that the oocyte and the sperm carry at the time of fertilization is very different. It actually reflects the very different history of the oocyte and the sperm that they had to undergo during gametogenesis. So the oocyte will mostly methylate sequences which are in two gene bodies during the process of oogenesis. So this is linked to gene transcription. While in the male germline, in the mature sperm, the sequences that will be preferentially methylated are intergenic sequences and transposon repeats. So de facto, at the time of fertilization, they will bring genomes with very different DNA methylation patterns. Not that these DNA methylation patterns will necessarily impact on the next generation, but they reflect their own identity of being an oocyte or a sperm. So of course, this creates a tremendous opportunity for differentially methylated regions that will be inherited by the embryo. So these are referred as germline differentially methylated regions. So most of the oocyte-specific regions that are methylated will be the ones that are in gene bodies, while sequences that will be more prone to be methylated in the sperm, as I said, are transposon repeats, where they will be mostly left unmethylated during oocyte growth. So if you apply a very crude technique to detect DNA methylation patterns in the embryo that has just been formed by fertilization, here using an antibody against methylcytosine, you can really clearly see the two sets of parental chromosomes. So these are the methylation differences that were directly inherited from the sperm and the oocytes, but also that have been exacerbated by some enzymatic activity just after fertilization. So here is the maternal set of chromosomes, here is the paternal set of chromosomes. So of course you can imagine that most of our sequences of our genome, so this is in mice, but that's the same in humans, will be differentially methylated on the maternal and paternal chromosomes, and that creates the opportunity of having mononuclear expression in the mammalian genome. If you apply the same technique later on during development, so here either after implantation or in adult tissue, you can see that you are no longer able to distinguish parental chromosomes by their methylation patterns using this same technique, because the tendency is to level any parental differences. So a sequence will be either fully methylated on the two parental chromosomes, and usually they will be repressed, or they will be now fully unmethylated on the two parental chromosomes, and they will be biallelic. Because of course you want to avoid as much as possible genes that will be only functional in one of the allelic copies. But there is one notable exception to that, of course. This is the case of genomic imprinting. These are specific sequences which have the ability to retain the memory from which parental chromosomes are coming from. And usually they retain this memory throughout life. So they are exceptional because, as we have seen during many instances during this day, the tendency of the genomes, as it's formed after fertilization, is to erase any information that has been inherited from the sperm and the oocyte in terms of DNA methylation. Because of course the embryo wants to be able to make any types of tissues, not only a sperm or an oocyte. And the DNA methylation patterns that it will inherit reflect a sperm or an oocyte identity. So there is first a wave of demethylation where you reach around 20% of methylation here, while there is 80% of methylation, 40% of methylation in the oocyte. And then after implantation and before the formation of all the different tissues, there is a remethylation of the genome, and then these methylation patterns are more or less stable throughout life. So the regions that are targeted for genomic imprinting, they are called imprinted control regions, ICR. And they are exceptional because they have the ability to maintain, as I said, their parent of origin methylation that they inherited from one of the parental germlines. So in the case of paternal ICR, These correspond to sequences which have gained DNA methylation during spermatogenesis, but not during oogenesis, and they will maintain methylation on the paternal allele throughout life, while the maternal allele will stay unaffected in terms of DNA methylation. And this will lead, as I said, to monolithic expression of a set of genes which are referred to as paternally imprinted genes because they inherit a paternal imprint. And then on the other side, there are the maternal ICRs, which acquire DNA methylation in the oocyte but not in sperm, maintain maternal specific methylation, and this leads to the monolithic expression of another set of genes referred to as maternally imprinted. So why are they exceptional? We know actually that there is a genetic determinism behind that. So it's often said that genomic imprinting is a paradigm of epigenetic regulation, but there is actually a genetic determinism behind that. As I showed you, there are like thousands of sequences which are inherited as differentially methylated from the oocyte and the sperm, but very few will be chosen to retain their parent of origin-specific DNA methylation. And almost all of them have been shown to carry the specific hexanucleotide motifs, TG-CC-GC, and when the internal CG is methylated, it will be recognized by a specific zinc finger protein called ZFP57, which is in abundance in the early embryo. And this zinc finger protein will recruit the CAP1 heterochromatic complex, and somehow, we don't know exactly what's the mechanism behind that, it will attract preferentially the DNA methylation machinery around these alleles that have this specific motif, which is inherited as methylated. And this is somehow how methylation can be maintained while the rest of the genome gets demethylated. Then why now the allele that is at risk now at the time of implantation is the one that is not methylated? How does this one is refractory to the wave of de novo methylation is not really known. It's thought actually that maybe transcription factors could also play a role. So for example, here, if these regions that is methylated in the sperm but not in the oocyte coincide with a promoter, an enhancer, if it's bound by a transcription factor, then it could be protected from the machinery that induces de novo methylation and somehow will stay protected during then the entire rest of life. So genomic imprinting is an intergenerational mechanism, but not a process, but not transgenerational. That's the best I could do for today. As I said, so differential methylation is established during gametogenesis. Then the differential methylation is maintained in the zygote, in the blastocyst, and later on. But then in the germline of this individual, there will be a mechanism of erasure of DNA methylation, because now you want to establish imprinting that reflects the sex of the individual in the next generation. So if this animal is a female, it will have both paternal imprints in somatic tissues, but in its own germline, it will have to erase anything to then re-establish only maternal imprints in the future oocytes. And then for if it's a male, it will re-establish only paternal imprints, paternal-specific DNA methylation in sperm. So how many of these regions are known? In the mouse genome here, that's roughly the same number in the human genome. So there are 20 known imprinted control regions. So that's a very small set of sequences. They control around 130 genes, which are referred to as imprinted genes. And you can see it's less than 1% of the entire set of mammalian genes. So that's really a minority. And here is a mouse karyotype, and these bars represent this ICR where they map, and you can see all the imprinted genes that are clustered around these regions because they are acting as cis-regulatory regions. And you can see that some of them will be maternally expressed and some will be paternally expressed. And this reflects actually the complexity of regulation mechanisms that act on imprinted loci to regulate monolithic expression. So one interesting thing is that we cannot really see the colors, but in red are the ones that are inherited as methylated from the oocyte. There are 17 known maternal ICR. So they are all intragenic. So remember, that's what the oocyte likes to methylate as sequences. And there are 3 known paternal ICR, and they are all intergenic. And this is the DLK1 GTL2 locus, DH19 locus, and the RSGNF1 locus and DH19 locus. But you can see that there is a clearly dominant role of the oocyte in controlling imprinted expression. So very few genes are subject to genomic imprinting, but they are involved in various diseases in humans. So either you can have mutations, so genetic mutations in these genes that will impair the expression of these genes, or you can also have epimutations that will also impact on the dose of these genes. So either they will become biallelic or completely lose their expression. So these are different syndromes that are associated with mutations or epimutations in imprinted genes. And in mice as well, it has been shown by targeted genetic studies that these imprinting genes tend to have functions in growth and regulation of development. So when you inactivate these imprinted genes, for example, you will get placental phenotype. You can get also fetal growth phenotype when you inactivate these different genes, or neonatal or postnatal growth phenotypes when you inactivate these other genes. And it's known as well they have a very important role on impacting on neurogenesis and behavioral control. So these different genes can affect neurogenesis, maternal care, sleep, memory, and so on and so on. So most of the studies that have been performed to infer the function of imprinted genes have been loss of function. So you completely abolish the function of these imprinted genes. Very few have actually addressed the role of imprinting in the function of these genes. So what's going on if now you turn them biallelic? Will it have an effect? So here it answers that these imprinted genes are essential, but why now if you completely abolish imprinting, does it matter? And then nobody has really looked into what will happen if you invert the parental origin of the imprint or the imprinted expression. Does it matter? So as I said, so there are like thousands of sequences. If you look at maternal genomic imprinting, which is the most dominant in the mammalian genome, you start with thousands of sequences which are only methylated in oocyte and not on the sperm. But if you look later on in any adult tissues, you only have 17 sequences which are able to maintain maternal specific methylation. So that's raised a question, are there many more to be discovered? Have we reached actually an equilibrium? Maybe we've identified them all. So a few years ago, we designed a screen to try to identify new maternal ICR to see whether this had been like just a tip of the iceberg or more would be to be discovered. So for that, we performed a DNA methylation comparison between embryos which have maternal and paternal imprints, so they inherit a fully methylated oocyte and a fully methylated sperm, so they are biparental embryos. And we compare them with embryos that are formed by fertilizing an oocyte that does not have any DNA methylation. That's possible because here there is a genetic deletion in one of the DNA methyltransferase cofactor, the MT3L, which is only required for germline methylation. So DNA methylation-free oocytes can be produced in mammals. So the oocyte doesn't need DNA methylation per se to be formed and to be fertilized. So you can fertilize this DNA methylation-free oocyte with a fully methylated sperm, and then you will produce maternal DNA methylation-free embryos. And we compared these two sets of embryos before these die. So they die at mid-gestation by placental defect. So we were like just upstream from the lethality of these embryos and compared their methylome using like a bit out-of-date technique now, which is MEDIP sequencing, where you pull down DNA methylation and perform high-throughput sequencing. And we then looked for sequences which would be differentially methylated between these two sets of embryos with the hope of identifying new maternal ICRs. So just to sum up, this is the design of the experiment. Sequences that would be only methylated in the oocyte and not in sperm. In these embryos, they will be scored as hypomethylated because they will lack this methylation. And so we found 45 regions which were hypomethylated in these mutant embryos compared to the imprinted embryos, which is a very limited number, as you can see. And among these 45 candidate regions, we had the 17 known maternal ICR, showing that the screen was efficient and selective. And we had then 28 potential new maternal ICR regions. So in red are the ones that were previously known. So here they are scored by their level of significance. And here are the new violet ones. And just to show you how a MEDIP-seq track looks like. So here's a wild-type embryo. You can see here the peaks of DNA methylation on this region. Here are these embryos that lack maternal imprints. And you can see they are like very similar to wild-type embryos, except specifically at these regions here, which coincide with a maternally imprinted genes, a known one, which is PEG12. So these new potential maternal imprinted regions were distributed throughout the mouse karyotype. There was no clear bias toward one chromosome or the other. And just to sum up what we found, we are not able to find any new canonical maternal ICR, meaning an ICR that will stay as maternally methylated throughout life in all tissues. So what we could refer them as lifelong and ubiquitous. So all the formerly known ICR were referred to be lifelong and ubiquitous, methylated on the maternal allele everywhere in all the tissues you can look at. But we found other forms of imprinting. Some of them were lifelong but tissue-specific, so that refer to sequences which will be inherited as methylated from the oocyte. But then in certain tissues, the paternal allele, which is hypomethylated in other tissues, will be regaining DNA methylation so that you wouldn't have any methylation differences in specific tissues while you still have paternal specific methylation in other tissues. So that's just to illustrate that. So for example, this gene, CDH15, happens to be maternally imprinted in the hypothalamus. You can see here all these maternal alleles that are methylated while the paternal alleles are not methylated, reflecting their status in the parental germline. So for that we used different mice with different polymorphism to be able to recognize the paternal from the maternal alleles. But you can see that in the other brain tissue, the cortex, there is no longer any parental differences because paternal alleles have regained the methylation during the process of differentiation of this specific brain tissue. So a lot of these new ICR that we identified were lifelong and tissue-specific, but all had in common to have the ZFP57 binding sites. And then we found even a more labile form of genomic imprinting, which we referred as transient. And this refers to sequences which also start as being differentially methylated with methylation inherited from the oocyte. But then at the time of implantation, during the massive wave of remethylation of the embryonic genomes, these sequences are not able actually to resist the genome-wide wave of de novo methylation. And we call them as transient imprinted clonal regions. So this showed to us that if there are new canonical maternal ICR to be discovered, they are probably very limited, so have probably reached what we have to know from now. But there are like other forms of imprinting which may not be as robust because they are not able to resist either microwaves of de novo methylation that occur during tissue formation and tissue specification, or massive wave of de novo methylation that occurs at the time of embryonic implantation. So we became a bit obsessed with this form of transient genomic imprinting because these sequences, they will keep differential methylation for very few days during mammalian development. So that represents 4 days during mouse development, like 1 week during human development. So what's the meaning of that? Why do you want to be differentially methylated for such a short region? So is it just an innocent byproduct of the huge amount of parental asymmetry that you inherit as an embryo, as a mammalian embryo, and then we'll have just some leftover until implantation, but that doesn't have any impact on the embryonic genome function? Or could it have an impact on the embryonic genome function, and could it have lifelong consequences, even though the existence of these forms of imprinting is very limited in time? So just to show what it reflects, so these are sequences which inherited methylation from the oocyte, not the sperm. If you look at the Bourc'his stage before implantation, most of the methylation is still on the maternal alleles, but then if you look after implantation, you can see that paternal alleles and maternal alleles are no longer distinguishable. And if you look at these sequences anytime during the life of this animal in any tissue, you have no idea that one day it was imprinted for a few days during development. And in terms of expression, that's also quite interesting to see that this will translate into monotic expression, a likely paternal specific expression if the ICR coincide with the promoter regions during pre-implantation development, but then this gene should be turned off for the rest of life, and you have no idea also that this gene was one day expressed during the first few days of development. So one paradigm for this form of transient imprinting is ZBF2, which is on chromosome 1. So it's a quite complex genomic locus. So just to sum up, it's on chromosome 1, as said, in mice. Here is the epigenetic status of the locus at the Bourc'his stage, so before implantation when it's still imprinted. This is the region that inherits oocyte-specific methylation. You can see here the lollipops are methylated on the maternally inherited allele, not methylated on paternally inherited allele. And then there is a block of repressive chromatin marks, polycomb marks, that stand over the canonical promoter of ZBF2. So ZBF2 is turned off. But these regions of differential methylation acts actually as an alternative promoter for a long form of ZBF2, which then will be expressed only from the paternal allele because the maternal promoter is obscured by DNA methylation. And what is quite interesting, as I told you, is a transient DMR. So as say, it will reacquire methylation on the paternal allele at the time of implantation, so there is no more parental distinction. But what we see in somatic tissues is the emergence of a new differential methylated region, an SDMR, as you can see. And this coincides actually with the disappearance of this block of polycomb on the paternal allele where there is DNA methylation. And this actually allows expression of ZBF2 from the paternal allele as well. So it's a very complex region. So genomic imprinting is in general very complex, but this one is, I think, on the top. But what is quite interesting is that here you have a switch from maternal germline imprint towards a paternal somatic imprinting. You can see the methylation switching from one allele to the other. And you have also a switch of promoters occurring from a long isoform of ZBF2 towards a shorter isoform of ZBF2. So we had two questions. First, what's the function of ZBF2? Why is it important to have such a regulation of the locus? And then, of course, what's the function of this short isoform that is under the control of the maternal germline methylation in the early embryo? So, first, ZBF2, where is it expressed? So we don't know what's the molecular function of ZBF2. It's a zinc finger protein. There is nothing much known about it. But what we found is that it's highly expressed in brain tissues and even more pronouncedly in the hypothalamus and the pituitary gland. A little bit testis. And using a different set of mice here with a LAX-Z reporter, you can see the very high expression in the hypothalamus here and the pituitary gland. So you may know that these two brain organs belong to the same axis and are responsible for producing a lot of hormones, including growth hormones, sexual hormones, and so on, to release them in the bloodstream of mammals. And ZBF2 is even more specifically expressed during the first weeks of life after birth. So in the pituitary gland and hypothalamus, you can see that both in males and females, the levels of ZBF2 mRNA will peak around the time of puberty. So ZBF2 is predominantly expressed in the hypothalamus-pituitary axis, and more specifically at the time of puberty. So we then went for a classical loss of function. So we disrupted ZBF2 protein at the genomic level on the paternal allele, because that's where you are supposed to have an effect, because here you should have a silent mutation if you do a genetic deletion, because there is no expression of ZBF2. And what we found is that there was nothing abnormal with these mice except a growth defect. You can see here some growth curves within the first days of life after birth, of postnatal life, and then you can see that this growth defect that you can see both in males and females will stay on until the rest of life for these animals. They are never able to catch up. They are smaller, and they are never able to catch up. So that's the only phenotype that they have. And as I said, you have a silent mutation. If you inherit the mutation from the maternal allele, you no longer have any differences in your growth rate. What we found is that this growth defect was specifically postnatal. There is no alteration of embryonic growth or fetal growth before birth. It really kicks in at the time of birth. It culminates at puberty. It's fully penetrant and it's systemic, meaning that all the organs are equally affected. If you weight the different organs of these animals compared to their wild-type counterpart, you can see that there is no differences when you normalize to the full body weight. And we can see that indeed the body weight is reduced, the lean mass is reduced, but there is no differences in bone area or fat mass. So it's a systemic growth defect. And what we found on top of this growth defect is a reduced survivability within the first days of postnatal life, meaning that ZBF2 is actually required for also postnatal survivability, and you lose almost like 50% of your progenies before P5 when you don't have ZBF2 being expressed. So just to sum up, because we need to know what could be the importance of ZBF2. So ZBF2 seems to be a positive regulator of growth, and which is also required for optimal viability before weaning. So, but then the next question was, of course, what is the role of this alternative form of ZBF2 being expressed in the pre-implantation embryos and under the direct control of sequence which is inherited as maternally methylated. So just to sum up, so LIS is expressed only during pre-implantation development and it's shut down as its promoter gets fully methylated and ZBF2 takes over. So the first model that we designed was a LIS loss of function. So we deleted the promoter on the paternal allele of LIS. So somehow we maternalized this allele because now you have a silent LIS on the two alleles. And we looked on the impact on the methylation and the expression of ZBF2 and also on phenotype. So what we found when we looked at these regions which is supposed to gain methylation on the paternal allele, we saw that there was no gain of methylation on the paternal allele, meaning that LIS seems to be required actually to promote deposition of DNA methylation on this secondary DMR. And what we found is that this was in the context also of a retention of this block of polycomb repression that sits over the ZBF2 promoter. You can see here it's an allelic assay for CHIP. Normally, you can see that most of polycomb sits on the maternal alleles here in red, and you can see that in the absence of LIS, you have the polycomb repression that is sitting equally on the maternal allele and the paternal allele where it shouldn't be normally. And this has an impact on ZBF2 expression, as you can expect. You can see here that there is no expression at all of ZBF2 when you quantify it in the pituitary gland and the hypothalamus of these mutant mice compared to their wild-type counterpart. So it's like having a complete null ZBF2, while the genetic sequence is completely normal. So, and in terms of growth effect, it was complete phenocopy of the ZBF2 mutation, even though here, as I said, the genetic sequence is the same, where these mutant mice that were not able actually to express LIS during pre-implantation development, they are also show all the same growth defect as the ZBF2 knockout, which are mostly postnatal growth defect. So this transcription during pre-implantation on the paternal allele is required actually to operate a switch so that now there is DNA methylation that is occupied here. This will dissolve the polycomb repression and allow ZBF2 expression later on during brain development after birth. And without LIS, you have exactly the same phenotype as having not ZBF2 being expressed at all. So then we wanted to investigate what would be the effect of imprinting. So LIS is required for establishing this epigenetic switch and to activate ZBF2. But what's going on now if you promote LIS on the maternal allele? So for that, we deleted the ZFP57 binding site that I told you about. So now we somehow paternalize the maternal allele so that we will have a double dose of LIS expression on both the paternal and the maternal allele. And we looked at the same molecular parameters and phenotypic parameters as before. And what we found is that now you have actually more DNA methylation that is deposited on the maternal allele where there shouldn't be any DNA methylation normally. And you can see it's not like a fully penetrant phenotype because we have like 60% of methylation that is gained compared to a full methylation that you find on the natural paternal allele. This is still actually associated with some dissolution of the polycomb group on the maternal chromosome, and this is associated with a higher expression of ZBF2 total, and also at the allelic level. You can see here ZBF2 being now expressed also from the maternal allele, where it is normally expressed only from the paternal allele in a wild-type animal. And what's the consequence in terms of phenotype of having now more ZBF2 being expressed in the postnatal brain? These animals are bigger, but only in males. The females are no different. So it seems that having expressed 2 doses of LIS will program ZBF2 to be allelic, and this will also program a bigger growth rate of these animals, which is also fully penetrant, the most obvious at the time of puberty, and as I say again, yeah, very penetrant. So then using these 2 set of animals, we could actually play with the parental origin of LIS expression and ZBF2 expression. So we crossed both LIS loss of function on the paternal allele and LIS gain of function on the maternal allele so that we could restore monadic expression of LIS during implantation development, but this time from the maternal allele. And we looked, can you restore a normal ZBF2 programming and another a normal growth programming? So here are different colors. That's a wild-type animal. That's an animal that has no Lees expression because it has a mutation on Lees inherited from the paternal allele. This is a Lees gain of function, so double dose of Lees. And this one has a monallelic expression of Lees, but this time from the maternal allele. And you can see that in terms of deposition of DNA methylation here at this region, that's the dose that you have in wild type. That's the dose that you have when you have no Lees expression. That's the dose that you have when you have biallelic expression of Lees. And you can see that you can somehow restore proper level of DNA methylation deposition when Lees is expressed only from the maternal allele. And then in terms of ZBF2 expression, you can see that normally when you have no Lees expression at all, you have no possibility to activate ZBF2 in the pituitary gland and the hypothalamus. And you can see that now by forcing Lees expression from the maternal allele, we can restore some proper ZBF2 expression level. And this impact actually on body weight, because these are the animals that do not have any Lees expression at all, double Lees expression and maternal Lees expression only, and you can see that they can somehow catch up with the normal weight and growth rate after birth. So at both the molecular and physiological level, it seems that Lees expression is completely able to rescue normal Lees paternal expression for programming ZBF2 and growth rate. Yeah, so what matters here is not really the parental origin of Lees expression, but more the dose that matters for growth programming. And here is actually 4 brothers coming from the same litter. We are lucky enough by breeding these 2 different mouse models. You can see here no Lees expression, double Lees expression bigger, and then maternal specific expression of Lees, and you can see that you restore proper body weight. And I remind you that these differ actually by doses of ZBF2 in the brain. But ZBF2 genetic sequence is not different at all. What matters is the epigenetic setting that these animals had when they were very tiny embryos. So here, if you have a monallelic expression of ZBF2, it has been determined by Lees expression on the paternal allele. Here you have biallelic expression because Lees was expressed from the 2 parental allele. Here you have etc., etc., sorry. And here we have like also proper setting of the locus, but on the reverse parental origin. So what it tells you is that even though some forms of parental transmission of epigenetic information may last for very few days during development, they may still be very impactful for the entire life. So here, I remind you the transient maternal imprint that we identified only lasts for a few days of development. It has no role for the embryo itself. The blastocyst is normal. It has no role also for any time point during pregnancy, but it will be impactful actually after the time of birth and will impact on the growth rate and also possibly on the survivability before weaning age. So why does it matter? It matters because this type of regulation may also happen in humans, and at least at the ZBF2 locus, we know that there is a very good genetic conservation, both at the synteny and genomic organization. Here is the mouse locus, here is the human locus. We know as well that the expression of ZBF2 is conserved in human tissues, so it's the most highly expressed in brain tissue, and specifically in the pituitary gland. And we know also that you have this switch of differential methylation between a germline DMR and a somatic DMR that is occurring at the locus. So this very complex regulation is likely occurring also during human development. So that raises the question as to whether maybe ZBF2 and the epigenetic control of ZBF2 may have an impact on height determination in humans. So height determination is a very complex mechanism. So we have started actually collaborating with Carol Huber to look as to whether there will be some SNP that will be associated first with the ZBF2 locus, and indeed there is one. And we are very much interested as well to see whether this very complex epigenetic setting of the locus that occurs during this very short period during gametogenesis and first days of embryonic development could be susceptible to maternal diet and of course to any kind of interference you may have on oocyte production and embryo culture. And I'm going to finish here. So the work I presented is mostly the work of a PhD student who recently graduated, Julianne. And we were very much helped by our transgenesis platform and also experts in pituitary gland development, which we are not at all. And I thank you for your attention.
On the same locus, Are there other imprinted genes?
No, it's a monogenic
It's on
imprinted
chromosome
locus.
2? It's
In humans,
not an
yeah.
imprinted locus area?
No, it's
No.
like a singleton. It seems to be a singleton. Yeah, we checked the imprinted status of the other genes around, and there was nothing.
With regard to the transient genes that show the difference just during a very short period of development, do you think that this could be some kind of a critical period where some environmental response would maintain the effect so it would last longer until adulthood?
Sorry, I didn't get your question. Are they more sensitive, you mean, or?
I mean, that they show that this difference only for a few days.
Yeah.
But maybe this is a critical period, and if under the right conditions, environmental conditions, this could be stretched longer.
I don't know. I have no idea. I think the regain of methylation that occurs at implantation is pretty robust. So unless you interfere with the wave of embryonic remethylation, which would be lethal for the embryo, I'm not sure. Yeah.
So you said you think we've found all the imprinted genes that are not transient. Do you think this is now sort of the beginning of finding tons of transient ones, or?
Yeah, I think, but will they be impactful? I'm not sure. So we've started playing with others. There was nothing spectacular. Okay. Yeah.
So, Deborah, maybe I missed it, but did you look at natural variation for imprinting across strains, and do you expect these 20 ICRs to be also the most invariant across strains?
They're pretty robust. So for the for the ZBF2 locus, we've also collaborated with the Icelandic cohort to see whether there would be EWAS loci. There was nothing obvious. But most of the EWAS analyses, they don't take into account the parental origin, where here they're just looking at differential methylation, but we need also to take into account the parental origin here. So, but yeah, they're quite robust, I think. At least among different mouse strains, or yeah.
Yeah. So, but
you still have. So what I didn't mention is that we found also some ICRs which were strain specific.
Yes, indeed.
Not among the robust ones, but like that the ones that are like tissue
indeed.
Specific, or yeah.
And so, do you think these ones could be sort of imprinted genes in the making, or is it reflecting specific life histories between?
So what's matter here? So why are they polymorphic? It comes back to transposons.
That's why I wanted to get you to get there. Yes, thank you.
So these correspond to loci where there is an LTR that is polymorphic among mice, and this LTR drive transcription during oogenesis, and if this ICR is on the path of the transcription initiated from this ICR, it will acquire methylation, and if it happens to have also ZFP57 binding site, it will act as an ICR later on. But yeah, I don't know if there will be, then you need selection to act upon it to see whether,
You said it was on chromosome 2 in humans?
Yes.
Yes. Are there any known uniparental disomy that would be associated with the phenotype of growth?
Not really, but the most striking phenotypic difference in terms of growth is really during puberty. And most of the assessment on height differences among human population is done during adulthood, so we may have missed that as well.
Okay.
Yeah.